We're going to move into talking about the mechanisms or the factors that affect the evolution of a group of organisms. But before we do that, we need to establish a few definitions so we're all on the same page about what I mean when I say some of these words. So, one of the largest misconceptions with evolution is that individual organisms can change or adapt on purpose. And I'm not saying that organisms in individuals don't change over time. We know that people change over time. You can change your behaviors, you can change the way you look, things like that. And so organisms, individual organisms, absolutely do change, but they don't change in a way that is heritable. Now, that's not to say that their gametes can't acquire mutations, because they absolutely can. But those mutations in gametes are not affecting the current individual. They affect the next population. So keep in mind that when we're talking about evolution, we're not talking about an individual choosing a particular trait or evolution acting on and causing a change in a specific individual. Evolution as a process occurs at the population or the species level. So evolution is working on a group of organisms. So at the highest level that we're going to talk about, we have species. And a species is a group of similar individuals that interbreed and produce viable offspring. So Homo sapiens, humans, are a specific species. We interbreed. As a whole, we produce viable offspring. The house cats are a specific species. They interbreed with one another, produce viable offspring. They are a different species than, say, a bobcat or a lion or an ocelot. Those are other species that can interbreed amongst themselves and produce viable offspring. Now, some people think, oh, you can get something weird like a liger or a tigon, and that's a cross between a lion and a tiger. The problem is that those organisms are themselves individual species, and they do not produce viable offspring. Within a species level organization, we can have populations. So populations are a group of individuals that are the same species that interbreed in a specific area. Now, populations might be fluid. They might flow into one another, or they might be completely isolated, or they might be somewhere in between. If we look at this picture on the right, we have two different caribou herds up along the border of Alaska and Canada. We've got the porcupine herd. They're indicated with the pink shading there, and their range goes from the water at the north about halfway down that peninsula there between Alaska and Canada. Now you can see that their range overlaps with that 40-mile herd, which lives a bit farther south. Now, some of these individuals that live farther north may never encounter the 40-mile herd and never interbreed. But it's possible, because their ranges overlap, that these populations might actually interbreed. Now, when populations interbreed, that allows them to exchange genetic information. And when they do this more regularly, the two populations remain more similar. If populations are isolated and they very rarely or never exchange genetic information, even though they're the same species, evolution is more likely to occur on one or both of those populations. When we get to chapter 12, we will talk about how that sort of isolation and different types of isolation actually lead to the development of new species. And then finally, evolution is actually acting on the gene pool within a population. So when I say gene pool, I mean all of the information contained in the DNA of all of the organisms. This is all of the alleles, all of the different genes, all of the alleles of all of the different genes. And evolution is actually working on that genetic information. So one of our definitions of evolution was that evolution was the change in allele frequencies, or the change in gene frequencies over time in a population. So as an example of what this means, we have these two tiger cubs here. We have a white tiger and the orange tiger. Now, let's say that you are a zookeeper and you want to increase the white tigers in your population. Now there's a couple ways you can do this. You can selectively breed white tigers. Let's assume that that white tiger is a female. You can breed her with a male, and when she has her cubs, she is going to have cubs that are white, and you have increased the relative number of white alleles in your population, and therefore decreased the relative number of orange alleles in your population. Now, the other thing you can do is acquire white tigers from other places. And when you do that, you bring white alleles into the population, increasing their relative frequency. So over here we have a pie chart. Now it doesn't matter which one of the examples we go with. Let's say that we bred this white tiger and the existing population in your zoo had this number of white alleles, just a real small fraction of the total alleles in your population. And then let's say she has, 2 or 3 tiger cubs. That's going to increase the number of white alleles in your population. Now remember, this is all out of 100%. So as the white alleles increase, the relative number of orange alleles has to decrease. Doesn't mean that the actual number of orange alleles decrease, but you've added individuals into your population. Now let's say that you continue your breeding program and you increase that white frequency of the allele. You have caused, over a couple of generations, evolution in that population. You have changed the number of white alleles, you've increased them in comparison with the orange alleles, and you have caused a genetic change in that population. Now keep in mind, a few minutes ago I said that populations evolve, individuals do not. Those white tigers, nothing changed about those white tigers, but you changed the composition of the population. So you have increased the frequency of the white allele in that population. So there are 4 major ways or processes that affect or cause evolution. So all of these change the, quote, market share of alleles in the population. This means that as one allele goes up or down, the other one changes respectively. And anytime that happens, even a little bit, you have evolution taking place. So we're going to talk about each of these 4 mechanisms in a little bit more detail. The first one is mutation. So remember that mutation is a change in the DNA sequence of an individual. And remember, the important thing about mutation is that it is the ultimate source of all genetic variation. So it is the only way that you get new information in a population. Now remember, we talked about some other things when we were in mitosis and meiosis and other ways that you can sort of reshuffle variation. But mutation is really important in evolution because it provides new material on which evolution can act. If you don't remember much about mutations, you can go back to chapter 6, section 9, and it talks about what a mutation is and some of the ways that it occurs. So a lot of what happens in the process of evolution is really sort of random events, and mutations are no exception. So mutations are random in the sense that we cannot predict ahead of time which individuals will get which mutations. So essentially, it is a coin toss whether or not one of your gametes is going to have a particular mutation. And it's a coin toss what gene or allele is going to be affected, if any. And then it's a coin toss as to whether the sperm or egg that you're producing actually gets that mutation. Because remember, in an evolutionary sense, mutations are only important if they occur in reproductive cells, because they're the ones that are passed on to the next generation of offspring. Mutations are also considered random because we can't predict if a particular mutation is going to be benign, meaning neutral, or not bad, not good, whether it's going to be harmful or whether it's going to be useful. So really, mutations are sort of in themselves a completely random event. And even if a mutation occurs, there's no way to know that it's actually going to affect the individual. So mutations tend to be the least impactful in the process of evolution, and that's because they're very rare. Remember that the process of DNA replication has some proofreading abilities incorporated into it. So DNA polymerase, our molecule that was copying our genetic information, can actually basically spell check for typos in the DNA and correct them as it goes. So the chance of getting a mutation during DNA replication is actually relatively low. And then even inducing environmental mutations is still pretty rare. So mutations are probably the least likely to be affecting the course of evolution of an organism, but we can't discount the fact that they can have a major impact on the organism or the population that we're talking about. So mutations are important because they can impact the fitness of an organism. Now, fitness doesn't mean that they're big and speedy and fast. Fitness means, and we'll come back to this, how likely an individual is to reproduce, to survive long enough to reproduce offspring. So if a mutation causes major problems in your ability to survive and reproduce, it's going to have a negative impact on your particular fitness. Likewise, mutations can be detrimental and cause early death, thereby reducing your reproductive success. Now, in general, if you look at any point in time, a group of organisms is going to be very specifically adapted to the environment that they live in. Now, they're not going to be perfect. They're never going to be perfect, but they're basically going to be the best they can be at that point in time. And so when you have a mutation, it very rarely is beneficial because you can kind of assume that that organism is already adapted to fit the environment that they live in. So any changes are probably going to negatively affect the individual. At the very least, they're going to have no effect on the individual. Having a mutation that actually increases their fitness is very rare. So on the right here, we have an example of a particular mutation. We've got our normal DNA sequence producing a normal protein that ultimately leads to the normal ear shape phenotype in this cat. Now this mutation is kind of a neutral one, especially if we're talking about house cats. This mutation causes a change in this image is indicating a single base pair, which probably means that it's producing a missense mutation and producing a protein, but that protein isn't correct. So it becomes dysfunctional. And then whatever this protein is not incorporated correctly into the physical structure of the ears, and you get these curled ear shapes in these cats. So this would be potentially the source of something called artificial selection, which we'll talk about later, which is where people are choosing traits that they like. So this curled ear, if you are a cat breeder, this might actually be a desirable trait and may actually artificially increase the fitness of these organisms. In the wild, it probably is going to be neutral because it's probably not causing much problems for these cats unless it's affecting their hearing or something like that. So our second mechanism of evolutionary change is genetic drift. Now the way that I remember this one is think about what the word drift means. It just sort of means to kind of go with the flow. And there's not really any controlled direction in a drift. So when we're talking about evolution, genetic drift means a random change in allele frequencies. So there are lots of things that can affect this, but generally genetic drift means sort of the random change in phenotypes or genotypes from one generation to the next. So let's say that you are a cat with the white patch of fur there on the left-hand side of that image. And as part of your population, some have the white fur patch, some Some have no fur patch. So when we're talking about genetic drift, we're usually talking about alleles that are not related to any sort of reproductive success. And therefore, inheritance is based solely on chance. So if we were to do a Punnett square of the individuals, maybe we would expect 1/4 of individuals not to have a patch, 3/4 to have a patch. So let's say that these two cats reproduce, and just by chance, you have more homozygous individuals without the white patch than you would expect, just by chance, because remember, each of those events is independent of one another, and it depends on which sperm fertilizes which egg. So now, just by chance, you have more cats without the white patch. Now this can change back and forth every generation. It depends on the cats, it depends on the genotypes, but one of the problems with genetic drift is that it can lead to something called fixation. Now fixation is the point where the population is made up of individuals with only one particular allele. Now this might not seem too important. We've got a picture on the bottom of how this genetic drift might happen and might lead to fixation. But the problem with this is that it reduces variation in the population. When alleles become fixed, there's no other alternative to those alleles, and this can be a problem because if you're talking about an allele that could in the future potentially give some sort of reproductive advantage, even if it's slight, and it's lost, then those organisms are less likely to be able to respond to a change in the environment or something like that. So variation is really important in evolutionary change, and we will talk about that a little bit later. But fixation can be detrimental to a population's ability to evolve in the face of changing environment. So here's another example of genetic drift. This is actually from a different biology book. Now, I like to say that these are plants in a meadow, and you'll see those red, pink, and white flowers. Now, if you follow the frequencies of the alleles on the bottom, the little P indicates the frequency of the red allele. The Q indicates the frequency of the white allele. Now, this is showing incomplete dominance, so we have a third phenotype in our heterozygotes, which is pink. This is very similar to the snapdragon examples that your book uses for incomplete dominance. Now, let's say that this is a lovely meadow in the mountains, and a moose walks by, and he walks down the middle of this meadow, and he just eats everybody in the middle, and then he comes back and he eats this guy. He doesn't care if they're red or white or pink. He's just hungry. So now those guys are gone. They can't leave their genes for the next generation. So only the highlighted ones are going to leave their genes. Now, if you'll see, you'll note, or if you'll look at the middle one, you'll notice that there are now more pink individuals in the population and less red. Now this is due to chance because that moose was not choosing any specific flower. He just happened to choose the one that was in his path. Now let's say that he comes through again and he's really hungry this time, and he gets almost everything down the middle, and he misses these two flowers on the edge of the meadow. What's left in the next generation is all red plants. So you can see we fixed this allele, and just by chance we have lost that white allele and therefore lost the white and the pink flowers. There are two specific types of genetic drift that I wanted to mention here. One is called the bottleneck effect. Now keep in mind that genetic drift is random with respect to the alleles that change in the population. So the bottleneck effect is where you have a very quick decrease in population size. Now this can be due to some sort of environmental factor. It can be due to a famine or disease that is not, none of these are specifically selecting the allele that you are looking at. And so after this bottleneck event, after the sudden shrinking of the population, you're left with a population whose allele composition is different than it was before. So here's our source population. For this particular gene, we have 4 alleles: blue, purple, yellow, green. There's suddenly a extreme and rapid environmental change. Maybe a massive forest fire came through the area and killed a whole bunch of individuals. And they were left with this small slice of the population. Now, just by chance, those individuals that were left only had the light blue and the light green alleles. Now you can see that the source population is much different from the remaining new population after this event. And they're sort of illustrating this bottleneck effect. Only some of the individuals survive and all of these other ones die. Now, this happened to cheetahs about 10,000 years ago. They believe that the cheetah population went through a population bottleneck, and the alleles for many of their genes became fixed. And when this happened, the resulting population was basically this set of cheetahs that were essentially all twins. They were all genetically identical, close enough that you can actually take a skin graft or an organ transplant from one animal and put it into the other, and they won't reject it because they're that identical. There's lots of other examples of this. This has happened to mountain lion population, or depending where you live, pumas or cougars, whatever you call them in the region that you're from. They've gone through a bottleneck effect. There's a Illinois prairie chicken that has gone through a bottleneck effect. This tends to happen when animals become endangered and their population quickly drops. Another type of genetic drift is called the founder effect. Now, the founder effect is when a small group of individuals leaves a population and establishes a new population somewhere else. And just by chance, those individuals have a different genetic composition than the original source population. So here's our original source population with a very small number of individuals with a blue allele here. Now, a small group of these guys leave, they establish a new population somewhere else. And just by chance, this allele is now more frequent in that founding population. So you have had a change in that population and the allele frequencies when that new population was established. So the example that our book uses is a disease or a disorder called polydactyly. And this is where you have more than 5 fingers or 5 toes. So if you just glance at those hands, they might just look weird. But when you look at them more closely, you can see that this woman has 6 fingers. This individual has 6 as well. He's got a real small thumb there that's sort of attached differently, and you can see the fingers are just generally off. Now, our book says that this has occurred in a high frequency in the Amish population in Pennsylvania, and that was because there was a husband and wife that founded that population that both had this disease. Now, this is a homozygous recessive condition. And so the resulting Amish population in Pennsylvania would have a higher percentage of this polydactyly allele than the source population that they came from, and also the rest of the human population around them. So our third mechanism of evolutionary change is something called gene flow. Now, gene flow is just a fancy word for migration. But remember that when we're talking about evolution, we're talking about specific alleles. So it is movement of the alleles that you're looking at into or out of the population. So when this happens, those individuals are probably not moving with respect to the allele that you're interested in. And so if you have a few individuals that all have that allele and they just happen to move into another population, they're going to change the allele frequencies in the population that they were in, and they're going to change the allele frequencies in the population that they go to. And so this is going to affect the relative allele frequencies in both populations. Now, again, this is sort of random chance because those individuals are not leaving because of those alleles. They just happen to bring those alleles with them.